Good afternoon, Professor Marwa. Maybe you can just come on and just confirm. Can you hear me okay? Okay, thank you. I'm not sure, can I hear you? Sometimes my speaker doesn't work very well. It's a pity after all these months that I'm still a little bit not so good with the technology, but we'll do our best. Okay. Professor Marwa, are you happy for me to start, or do you want to introduce me? No? No, I can't hear you. Could somebody just come online, please, to just make sure that I can hear you? If you can just say something, and then I can be sure that I hear you. Yes, so I'm not able to hear you. If somebody is talking to me, unfortunately, My speaker doesn't seem to be working as well as I would hope. Look, I suppose the most important thing is that you can hear me, if that's okay. Okay. Look, what I'll do is I'll probably just push ahead there. I said I'm not sure if somebody's trying to talk to me, but I can't hear anybody. So look, I'll just push ahead and I'll start in with my lecture. Look, I guess just to introduce myself, my name is Owen Fleming. I'm a lecturer in biochemistry here in University College Cork. I'm linked with, I suppose, both the School of Biochemistry and Cell Biology and also the School of Pharmacy. So that's why I'm talking with you now today. My name is spelt a little bit weird in that here we have it's E-O-I-N, so I'm not sure how well you would pronounce that in Egypt, but it's pronounced as O-W-E-N. So Owen Fleming is my name. Okay, so look, what I'm going to do today within the next 45 minutes or so is I'm going to talk to you a little bit about some of the principles associated with recombinant DNA technology as part of your PMI 525 course, which is pharmaceutical biotechnology. And I suppose, look, just from the start, we know from lectures that you'll have done previously that genetic information is stored in the form of double-stranded DNA. And the example that I have here on this slide is for eukaryotic cells, but it's not that dissimilar for prokaryotic cells, for microbes, for bacteria, where again, the genetic information is stored in the form of double-stranded DNA. Now, specifically in relation to the eukaryotic cells, this DNA is packaged very, very tightly in these things called chromosomes, of which there are a total of 46. But along each one of those chromosomes, what we find is that there are multiple protein encoding genes. So here, for example, we might have a DNA inside in a nucleus, or in the case of bacteria, just free present in the cytoplasm. And if we look along this length of double-stranded nucleic acid, if we look along this double-stranded DNA, we can identify a number of different regions that we would identify as encoding specific protein encoding genes. So I guess the molecular logic for life, which is what we call it, and this is similar for both prokaryotes and eukaryotes, is that double-stranded DNA, and specifically the nucleotide sequence within the double-stranded DNA, gets transcribed into a nucleotide sequence in single-stranded RNA. And the process here that Dr. Gahan will have spoken to you about last week, this is called transcription. And then that single-stranded RNA is then converted into a specific amino acid sequence in a polypeptide chain. And if the polypeptide chain is long enough, we often refer to it as the protein. And I suppose the key thing here is that the specific function associated with that particular protein is a reflection of the specific nucleotide, as the specific amino acid sequence in the polypeptide chain. And it is that specific primary sequence that allows for the polypeptide chain to fold into a specific three-dimensional structure that will be held together by a variety of non-covalent bonds, such as hydrogen bonding, van der Waals forces, hydrophobic interactions, and ionic interactions. And in all cases, that amino acid sequence, that protein function that is derived from the amino acid sequence, is reflected by a nucleotide sequence. So there's a clear link between nucleotide sequence in DNA and amino acid sequence, which then gives rise to a specific function linked to a protein. So are there situations where we would want to make protein? And yes, now that we understand how genes are expressed in cells, there are situations where we wish to apply this knowledge in order to make proteins, okay? So what might these advantages be? Well, certainly from a research point of view, if we can get proteins, or we can look at the expression of proteins within cells, it gives us a better and a deeper understanding of the function of those proteins. And I suppose it also gives an insight as to what happens if something goes wrong, if a cell begins to develop a disease. This is more often than not occurring as a result in the change of some sort of a protein function, which in turn may result as a consequence of a change in the nucleic acid sequence, or the nucleotide sequence, that we find inside in the DNA. So yes, from a research point of view, from a pharmacy perspective, from a medical, from a health perspective, we are interested in being able to make proteins, convert DNA and the nucleic acid sequence, the information in the nucleic acid sequence, into a protein which has a specific function. And once we have studied the protein in the context of the cell, once we've looked at the protein in the context of disease, we're now in a position maybe where we can begin to intervene, or begin to think about developing therapies that we can use in order to treat disease. So one of the big interests now that we have, and certainly from the point of view of your pharmaceutical biotechnology course, we have a key interest in terms of making proteins, protein drugs that have specific functions that we can use in order to try and treat disease. So yes, we are interested in, I suppose, manipulating and creating a situation whereby we can turn DNA into protein. And really, this is what I'm going to talk about now today, is this idea that inside in our DNA, whether it be maybe a bacterial gene and a bacterial protein, or a eukaryotic gene and a eukaryotic protein, we have an interest in identifying genes that code for proteins, and then possibly getting that nucleic acid sequence to be converted into a protein that then has some sort of a function that we can explore, and that we can research, and we can examine, and possibly manipulate in order to treat disease. So just, I suppose, as a very general overview in this slide and the next slide, and I know that Professor Gahan will have spoken to you about this in passing last week, but in terms of our ability to turn nucleic acid into protein in a controlled laboratory type of context, there are a certain number of tools that we need in order to be able to do this. And what this slide just shows is two of the critical tools that have been used, or that are being used. And they were originally identified actually in bacteria. So these are examples where, through our increased understanding of what goes on at a molecular and cellular level in prokaryotes, in bacteria, we can then take that information and begin to apply it in a context that applies to eukaryotic cells, applies to humans, and we're going to see how it can be applied in order to treat disease. But the two key elements of the bacterial physiology, I suppose, that we're interested in, involves plasmids and restriction enzymes. So plasmids are circular DNA that are found in bacteria. Here we have the chromosomal DNA for the bacteria, and here out in the cytoplasm, we have this circular double-stranded DNA. And these are, I suppose, they have evolved over millennia in bacteria, and very often they confer an ability on the bacteria to express some extra proteins that are not present in the bacterial genome. And these proteins may have special functions that allow for some sort of selective advantage for these particular bacteria. So historically, these are native to bacteria, and it was, their discovery and our I suppose, characterization of them that now allows us to use them in terms of biotechnology. The other component of the bacteria that we're interested in are restriction enzymes, and this is shown down here in this lower diagram as this green kind of protein blob that is binding to the double-stranded DNA. And what you can see is that the restriction enzyme, it's recognizing a particular sequence in the DNA, and it is cutting that sequence. So again, this is something that has occurred naturally in bacteria. We see them across a variety of different bacterial species. And they're, I suppose, evolved to try and maintain or to prevent viruses, bacteriophage, from coming in and infecting the bacteria. So this is a way for the bacteria to cut up foreign DNA into short pieces. All right, so these are the key tools now that we're going to use, and we're going to use these in the context of recombinant DNA technology. And the specific aspect of this recombinant technology that I want to talk about is often referred to as a kind of a general term of gene cloning. So gene cloning is the process of making multiple identical copies of a particular gene. The cloned gene then can be manipulated and put into an appropriate cell, and that cell will then be capable of making the protein that is encoded by that gene that we cloned. Okay, so this is, I suppose, just a summary and overview of it. In relation to the gene cloning, a gene is inserted into a circular piece of DNA. This is our plasmid, and here is the gene that encodes the nucleic acid, the nucleotide sequence, the double-stranded DNA that codes for a particular gene of interest. So we can now take this gene of interest and we can put it into the plasmid, recreate this circular plasmid, which we can put into bacteria through a process referred to as transformation. So once we have this circular DNA, once we have this plasmid, through the process of transformation, we can take that DNA molecule, which when we manipulate DNA in this way, we often refer to it as recombinant DNA, and we can put this into the bacterial cells. So through a process of transformation, which often involves heat shocking the bacteria, we can get the plasmid to go into the cell. And here we have a situation where we have the plasmid that contains our gene of interest, now located within a cell that's capable of producing the protein that is encoded by that specific genetic sequence. In this scenario, as the bacteria reproduce, as they divide, they replicate, and the recombinant DNA plasmid molecule also replicates and gets passed on to their offspring. So in this way, gene cloning yields multiple copies of a gene that's present in all of the bacterial, I suppose, offspring derived from that original cell that took up that plasmid. So a little bit more depth now in relation to these specific molecular tools that we're going to use in order to achieve gene cloning. And the first one that I'm going to talk in a little bit more depth about is these restriction enzymes. So there are three classes of restriction enzymes. The ones that we are particularly interested in are referred to as type 2 restriction enzymes. And these recognize specific, what are referred to as palindromic nucleotide sequences in DNA. Once they recognize these sequences, these enzymes are endonucleases, so they're capable of cutting nucleic acid. So they cut both strands of the DNA molecule. And they do so by recognizing as a specific palindromic nucleotide sequences, and depending on the restriction enzyme, they will be capable of recognizing sequences that could be 4 nucleotides long, 5, 6, 7, 8, that the specific recognition sequence varies depending on the specific restriction enzyme. So these palindromic sequences, a palindromic sequence in DNA is one in which the 5' to 3' base pair sequence is identical on both strands. So here we have our double-stranded DNA. Let's say, for the sake of argument, that this is 5' to 3', and we know that there is this directionality associated with the nucleic acid strand. The complementary strand then is 5' to 3'. And what we see here is that the 5' to 3' nucleotide sequence on what we call the sense strand, GAATTC, is identical now to the complementary strand in the opposite direction, GAATTC. C. So in terms of the restriction enzymes, in terms of their names, this enzyme EcoR1 is an example of a restriction enzyme. HindE3 is an example of a different restriction enzyme. And what we're going to see here is that the names are given to these enzymes in a fairly systematic manner, in that the E stands for Escherichia. The CO part refers to coli, which is the species. R is the specific strain that the enzyme was purified from. And then the number refers to, this is the first endonuclease isolated from this particular bacterial strain. HindE3 then is a second type of restriction enzyme. Again, it goes through here the different letters that are used in order to name the specific restriction enzyme. And you can see here now, excuse me, over on the right-hand side, the specific palindromic sequences where these restriction enzymes are capable of recognizing and cutting the double-stranded DNA. So you can see here for the EcoR1 restriction enzyme, it's GAATTC, and you can see that there's this kind of internal mirror reflection going on between the sense and the antisense strands. Again, for HindE3, the recognition site is AAGCTT, and again we're seeing, you know, that the sequence is identical, going 5' to 3' on either of the two strands. So it says these are examples of the restriction enzymes now that are so critical to our whole gene cloning enterprise. And I suppose the other characteristic thing about these type 2 restriction enzymes now is that not only do they recognize specific sequences with this internal palindrome in them, but they also cut the double-stranded DNA, or at least some of them are capable of cutting the double-stranded DNA in such a way that it creates a kind of a staggered cut. So rather than cutting the double-stranded DNA right down the middle of the recognition site, it cuts it in a staggered way. And to give an example of this now, we're going to come back here to our EcoR1 restriction enzyme. And what we see is that when this EcoR1 restriction enzyme recognizes its nucleic acid recognition site and it cuts it, it does it in a staggered manner, so that it digests one strand here between the G and the A, and it cuts the other strand between the G and the A, which means that it creates this sort of a staggered product. Okay, and these short staggered tails are often referred to as cohesive or sticky ends. Okay, and the reason will become clear why that's the case, because this is kind of like a sticky hook that can find other compatible hooks in other nucleic acid sequences. And we'll be talking about this and the importance of this when it comes to gene cloning in a moment. So the example here is EcoR1. It gives rise to 3' overhang. Other types of overhang vary depending on the restriction enzyme. So for example, the EcoR1 gives rise to a 5' overhang. BAMH1 gives rise to a 5' overhang. Other restriction enzymes, such as PST1, gives rise to a 3' overhang. But again, all the time, the key thing that you need to be aware of here in relation to this is the concept that these overhangs act as sticky ends, or they can act as hooks in order to identify and interact with compatible sticky ends on other nucleic acids. So when we have a length of double-stranded DNA and there is a specific restriction enzyme that's going to recognize a palindromic sequence, it's going to recognize and it's going to cut that palindromic sequence. It's going to maybe do it in the staggered manner that you see here on the overhead or the slide in front of you. And by so doing, it releases this internal region, this nucleic acid region, between the two restriction enzyme digestion sites. And what I'm trying to communicate now in terms of this particular slide is that when we do this, when we take a long stretch of DNA, maybe your genome, for example, and we add a restriction enzyme that recognizes specific restriction sites, target sites in the nucleic acid and cuts them, we can then isolate those cut, or what we call those digested fragments. So we can do this by running the DNA out on a gel. And depending on the size of the fragment that is released, it gives rise to shorter or longer fragments on an agarose gel. And it is possible for us then to remove that band, a specific band of a specific size that corresponds to a specific product arising from the restriction digestion of a fragment of DNA. We can isolate that fragment. So in effect, we can isolate the nucleic acid sequence, the double-stranded DNA that codes for a specific gene. So we now have this technology where we can get the DNA from a source. In this case here, I'm talking about a human nucleus. We isolate the chromosomal DNA, which has individual genes located along the individual chromosomes. And we can use restriction enzymes maybe to isolate or to cut out the nucleic acid sequence that corresponds to just one of these. Okay, so now we have the nucleic acid sequence that codes for a human gene. The question is, if we want to make the protein that is encoded by this gene, how are we going to take it and put it into a cell in order to get that cell to make our protein of interest? So in principle, yes, we can take just the nucleic acid sequence that codes for a protein and just put that nucleic acid sequence into a cell. But look, the bottom line is it's really difficult to put one of these into each one of these individual cells. And once it goes into the cell, there's nucleases and stuff present that are going to break it down. So the expression of the protein will be time limited. So how do we overcome this? Well, we overcome it by exploiting one of those key features of our restriction enzymes, in that when restriction enzymes recognize a particular site in the nucleic acid sequence and cut it, creating characteristic overhangs, sticky ends, this can then, I suppose, in many respects, act as a hook to bind to compatible cohesive ends in other pieces of nucleic acid. So it doesn't need to be the original chromosome, for example, that the gene has been cut out from. It can be any nucleic acid sequence, as long as there are these compatible sticky ends, then our fragment of interest, the nucleic acid sequence that encodes for our gene of interest, is capable of associating with some other nucleic acid. And this is where our plasmids come in. So plasmids can act as vectors in order to carry genes into cells, in much the same way that plasmids are used in order to carry genes into bacterial cells. So for example, a gene that might code for antibiotic resistance that is incorporated into the plasmid DNA, that plasmid can go into bacteria, and actually it can move between bacteria as well, but it can go into bacteria. And in so doing, it carries that nucleic acid sequence. It is a vector for carrying the gene into the bacterial cells. So plasmids are molecules of DNA that are found in bacteria. And in terms of the specific features of plasmids that are required in order for us to be able to use them in this capacity, in terms of utilizing them for biotechnological applications, there's these features. One of the first ones here is an origin of replication. So what happens is that when we put the plasmid into a bacteria, such as is happening here, there is an origin of replication, so that when the bacterial genome is being replicated, the plasmid DNA, the circular DNA, also becomes replicated. And that means that in the daughter cells, when that splits into 2 bacterial daughter cells, they will also continue to have the plasmid in them. So that's an important feature. Second really important feature is having unique restriction sites. So there is within the plasmid what we would call the multiple cloning site. And the multiple cloning site encodes the nucleotide sequence for lots and lots of different restriction enzymes. And these tend to be restriction enzyme recognition sites that are not present at any other site within the plasmid. What this means is that we can use specific restriction enzymes such as EcoR1 in order to cut open the plasmid. And in so doing, which is shown here on the left-hand side, it creates characteristic overhangs to which sticky ends from a gene that has compatible EcoR1 sticky ends, it can essentially go in and bind to that sequence using base pairing. So the next feature of plasmids that makes them really good in terms of acting as vectors to carry genes into cells, well, they're a relatively small size, which means it's comparatively straightforward in order to get the plasmid to go into cells. And this is shown here in the context of the plasmid going into bacterial cells through the process called transformation. But a little bit later, and next week, I will be talking to you in a bit more detail about how we can put plasmids into other types of cells. So we can put plasmids into mammalian cells through a process. It's referred to as transfection, but it is pretty much similar to what transformation does, putting plasmids into bacteria. So this is another key feature of plasmids. We can also insert into the plasmid the nucleotide sequence that encodes for proteins that confer selectable markers onto bacteria that have the plasmid. So the key example here is that we can include the nucleic acid sequence for proteins that confer resistance to antibiotics. And what this means is that we can grow the bacteria now in media that contains specific antibiotics, and the only bacteria that are going to grow are the ones that have the plasmid and have this selectable marker inside them. But it says there's a number of other examples in relation to this: tetracycline, chloramphenicol, and specifically for ampicillin, this is the molecular basis for the resistance that it confers. So in effect, what has happened is that through the decades since plasmids were identified, that naturally occurring plasmids have been engineered and modified to possess these key features and make them useful as cloning vectors, okay? So, for example, this plasmid here, PUC18, has a number of restriction enzyme potential cloning sites that we can cut the plasmid open, and we can insert a gene of interest into the plasmid that we want to express in cells. So this is that process. Here we have DNA, we have the plasmid. We isolate it from bacteria. We digest it using restriction enzymes. We then take, DNA that contains our gene of interest. For example, this could be a chromosomal DNA from bacteria or from mammalian cells. And we cut it so that we can digest the specific target gene that we're interested in and isolate it. So we would isolate the plasmid, we would isolate the gene target of interest. We would do this using agarose gels. And then we use an enzyme called T4 DNA ligase that catalyzes the formation of phosphodiester bonds. And if you recall, this is the specific type of chemical bond by which individual nucleotides are covalently bonded to each other along the double-stranded DNA. So here we have TTCGA. So the bond that holds these together is a phosphodiester bond. Now, what happens here is that we have our target gene here located within a longer section of DNA. We use restriction enzymes in order to cut this long double-stranded DNA to release the specific fragment that we're interested in. And now, using these sticky ends, these compatible cohesive ends, these will interact with the compatible sequences in the digested plasmid backbone. So this interaction is non-covalent. So we're talking about the interaction between A and T, between G and C, between C and G, between T and A. So we know that this involves either 2 hydrogen bonds or 3 hydrogen bonds, depending on the base pairing between specific nucleotides. Once this non-covalent interaction has occurred, we can then come along with the enzyme that catalyzes the permanent covalent bonding of this insert into the plasmid. So this is what's happening here. We can then transform that into bacteria, and in the presence of specific antibiotics, we can select for the plasmid or the bacteria that have specifically taken up the plasmid. Now, all of this works fine, except when you think about it. Sometimes the pieces of DNA that we want to clone may not have the appropriate restriction enzymes located at either end of the fragment that we want to clone. So, these restriction enzymes and recognition sites can occur at any point within the genome. So sometimes they're located within genes, located outside genes. And in this kind of scenario, even though ideally we would like to be able to cut our human DNA, liberate our specific fragment, this one here, we're liberating this fragment, and we're trying to clone this into the plasmid using T4 DNA ligase. Quite simply, this cannot happen in practice. Okay, and it's only because of this, I was going to say the random, but it's evidently not random, but this localization of restriction enzymes within double-stranded DNA. We don't control that. At least, within the genomes, we are unable to control it. However, what I'm going to talk about now for the next few slides is this process of polymerase chain reaction that I'm sure you've come across previously, but how we can utilize this particular technique, this technological approach, in order to create fragments of double-stranded DNA that contain the restriction enzymes that we're interested in using in order to clone into a particular plasmid. So just to review this a little bit, for you, in terms of replication of DNA in the genome, we know that a fragment of DNA can be replicated using the enzyme DNA polymerase, using individual nucleotides, and a short primer. Okay, so what happens now in terms of what happens in our cells is that there's a short primer. And going in a 5' to 3' direction, the DNA polymerase adds 1 nucleotide at a time in order to create a double-stranded DNA. Okay, so this will create the double-stranded DNA, and also there will be another strand here. So the process associated with replication is that there is separation of the 2 strands, there's complementary priming, and then DNA synthesis. So this is what happens all the time when cells divide. And there was a really intelligent guy, I suppose about maybe 30, 40 years ago at this stage, who figured, maybe we could exploit this in order to make lots and lots of copies of a specific DNA sequence. So this is the approach that he came up with. It's referred to as the polymerase chain reaction. And, I suppose it really isn't an understatement to say, in terms of technological advancements, PCR represents such an incredible opportunity and represented such an incredible opportunity during the last 30 or 40 years that it has transformed the situation in terms of our understanding of science, in terms of our understanding of disease. It is the basis for sequencing of the human genome, for example. It is also the basis for the qRT-PCR tests that are currently being used in order to identify individuals that are infected with COVID, with SARS-CoV-2, and have COVID-19. So this is an incredibly important technique, and allows for both strands of a DNA fragment to be simultaneously replicated using DNA polymerase, nucleotides, and short DNA primers. So this is what goes into the PCR reaction tube. And just like the replication inside in our cells, I suppose the experimental parameters are manipulated so that there is separation of the strands of the double-stranded DNA. And this is achieved using heating. So this is the denaturing step. Then primers, which are complementary to specific nucleotide sequences. Primers go in and bind very specifically to the complementary nucleic acid sequence in a target fragment of DNA. And then at 72 degrees, which is the temperature at which the special thermoaquatic or thermostable DNA polymerase activates or is functioning, we get the extension, we get the elongation of the replication process in order to create double-stranded DNA that is an exact copy of the original template DNA. So this is our process of polymerase chain reaction. And I suppose the thing is, if once you've completed one cycle, if you then increase the temperature in order to denature the strands, if we get annealing of the primers to their complementary sites, and then an optimal temperature at which the thermostable DNA polymerase is going to function, we're going to get a repeat production of an amplified fragment identical in sequence to the original template. So through this process, by increasing the number of cycles, we can increase exponentially the amount of DNA that's produced. So what we can do now is we can perform PCR and create DNA fragments, PCR products, that contain restriction enzyme digestion sites at the ends. So here we have a primer, which as part of the PCR amplification is going to bind to compatible sequence and begin that extension. But when we design the primers, we can include a part here at the end that initially is not complementary to the target DNA, but it contains the restriction enzyme recognition site for a particular enzyme. What this means is that when we create, excuse me, when we create these PCR products, now, not only does the amplified fragment of DNA contain an exact copy of the original sequence that was used for the template, but it additionally contains nucleic acid sequence at either end that is going to act as the recognition site for restriction enzymes. So this now is our PCR product. We can take the PCR product and we can put it into a restriction digestion, which gives rise to these sticky ends, these compatible cohesive ends, and this can then be cloned into a plasmid. So here we have our PCR product. Digested with the restriction enzymes, we know now that this particular nucleic acid sequence contains the entire coding sequence for our gene of interest, and it contains restriction enzymes at either end that are unique. There's no internal restriction enzyme recognition site. The only restriction enzyme recognition sites are going to be at either end. Now, what we can additionally see is that if we use two different restriction sites, in this case Hindi 3 and down here, echo R1, it means that when we now want to take this DNA of interest and clone it into a plasmid that has been prepared using the same combination of restriction enzymes, we can get that DNA fragment to go in a specific direction. And that's what we need, because we need for that nucleic acid sequence to be transcribed into RNA and translated into protein. And if it goes in back to front, then it's a different nucleic acid sequence, which gives rise to a different amino acid sequence, which will give rise to a different protein. So by using this system where we create a PCR product, DNA is amplified and is cleaved by the same restriction endonucleases that are going to be used to produce the sticky ends in the plasmid, we can then use the T4 DNA ligase in order to clone that fragment, transform it into a bacterial cell, and grow up the bacterial cells, selecting specifically for those that have taken up the plasmid. So now once we have the plasmid, we can put it into a production organism, which is going to produce a protein of interest. Now, very often we would do this, we would use this in the lab as part of research-related experiments. But again, from a biopharmaceutical technology point of view, we may be interested in getting organisms that produce this protein inside in cells. So, and then purify it from the cells. So we can put plasmids into bacteria, we can put it into yeast, we can put it into mammalian cells. There are different advantages to different cell types expressing different types of our different proteins. For example, a lot of the drugs that are produced now, protein drugs, they're made in eukaryotic cells, in mammalian cells, and specifically in Chinese hamster ovary cells. And the reason for this is that the proteins undergo appropriate post-translational processing, such as glycosylation, for example, that are essential for the functional activity of these specific protein drugs. Okay, so that's the end of my my presentation now today. Does anybody have any questions? We have, I think, 2 minutes left there. Is there any questions in relation to any of that material? No. Okay. So look, I think I'm due to go and present now to the other group. So if there are no questions there, what I would just say is thank you for coming online and listening to me now for the last 45 minutes. Please feel free to email me. You know, Professor Marwa has my email address if you have questions in relation to some of that content. And I look forward to meeting up with you now next week, and we'll take it a step further, and we can, I suppose, take account of some of the applications of this in terms of producing engineered proteins that are appropriate for the biopharmaceutical industry. Okay, so thank you very much. Have a good evening there. Thank you.